So now I invite Professor Abhay H. Pande and to deliver the talk on the engineered proteins. Dr. Pande is an associate professor in the Department of Biotechnology in NIPER and having the 22 years of experience in the field of the protein research. And he has graduated from the Nagpur University and PhD from the DAV Indore and postdoctoral experience. So, Dr. Pande, thank you.
Thank you. Good morning. Thank you very much, sir, for my introduction. Today I will talk about engineered proteins, a new era of biopharmaceutical innovation. As we all know, biopharmaceuticals, they are derived from biological source. They offer many advantages over chem pharmaceuticals. Just to bring a point to the importance of biopharmaceutical, last 4-year survey shows that out of 207 products approved by US FDA between January 2014 to 18, 97 products were biopharmaceuticals, which constitute around 47% of approval. This is higher than 20% in 2010 survey and 26% in 2014 survey. So the point here is everybody know and agree that biopharmaceuticals are coming in a very high demand. And when we look at the biopharmaceutical per se, we'll find that majority of segment is occupied by very important molecule, monoclonal antibodies, followed by contribution from other biomolecules. So this is a scenario of biopharmaceuticals. Can I have next slide, please? Okay, it's fine. So coming back to protein biopharmaceutical particularly, we can classify them into 3 broader categories. We all are aware of these terms, biosimilars. They are similar but not identical to the innovator molecules. They differ in very many different aspects. They are called by a variety of names, follow-on biologics, biogenerics, similar biotherapeutics products, generics biologics, but they are biosimilars. They are still in high demand. As per latest survey, 89% of the approved drug taken by the human population, 89% were generic in nature, either chem generic or biogeneric. Then there is a second category called biobetters. They are nothing but the upgraded version of the innovator molecule. They are also called by a variety of names, and it is one of the fastest growing segment in the biopharmaceutical market. So this is what we are looking at it, improving or upgrading the proteins which are either approved or under advanced pipeline. And then comes the third category, which is a novel or innovator molecule. We know it takes long time and huge money to develop a novel innovator molecule. India is called the pharmacy of the world. It's a leading source of path-breaking innovation, our biopharma industry or our pharma industry. And major segments are generic drugs, API, intermediate, drug formulation. Recently, we have proven our strength in the field of vaccines. And we also have biologics and other segments. My question is, can we make a protein biopharmaceutical another major weapon in our arsenal? Please remember, our Pharma Vision 2020 exclusively says to make India a major hub for end-to-end drug discovery. So keeping all those things in mind, biobetters or improving the property of existing drug can be a lucrative segment. There are very many limitations of the biopharmaceutical or protein drugs. Example, suboptimal pharmacokinetics. Their circulatory half-life is less. So we have to take it in infusion form or frequent dosing. Another limitation is suboptimal pharmacodynamics. We can increase the efficacy of existing drug. Poor blood-brain barrier permeability is another area where we can look into protein engineering and develop a molecule which, if you want to deliver it across the blood-brain barrier, and the target specificity can also be improved. So by using protein engineering, one can address these properties, one can implant these properties to already existing drug and make them bio-better. So chimerization of existing or pipeline drug by protein engineering can address these limitations, and biopharma across the globes, they realize this importance and they are already in process of that. One of the example I can take improving the target specificity is a chimeric streptokinase drug, clot-specific drug developed by IMTech, Dr. Sahni, where we have generated the chimera molecule, and this drug is in the phase 2 trial last reported. So by using protein engineering or chimerization, one can develop a target specificity in the molecule. This can be an example that we can improve the blood-brain barrier permeability by engineering a chimeric protein, and Armagen has developed a platform technology to deliver protein to the brain, which can cross blood-brain barrier. Chimerization can also improve the PG properties. For example, Ablynx has developed trivalent antibody, which has a higher affinity or value as compared to the bivalent monoclonal antibody approved by the authority. So this can be an example that you can increase the efficacy of molecule by engineering, and finally one can improve the pharmacokinetics property as well. So this is the area which I am going to talk about in the coming slides. Many proteins, they have low circulatory half-life, as a result of which we have to take it at the higher doses or frequently or administered in the form of infusion, and that results in a lot of issues, higher prospect of adverse reaction, cost of treatment, and financial burden. Thus, developing an upgraded version of protein therapeutics having increased circulatory half-life is the need of the hour. And chimerization can improve the pharmacokinetics property of protein drugs. For example, GLP-1R agonist market is very huge. It is used for glucagon-like peptide 1 receptor antagonist. It is performing well in T2 diabetes. The market is huge. And as we can see, by protein engineering, you can increase the half-life and provide some benefit to the patient. Earlier it was used, some formulations are twice daily, then came the once daily, then by chimerization came once weekly, and people are going further that can be developed a still long-lasting GLP-1 analog which can be used for these populations suffering from T2 diabetes. Another example is factor 9, which is used for hemophilia B. So protein engineering by chimerization is being used to increase the half-life from 17 hours to more than that, 82 and 102, and more engineering of protein is going on. So this tells that by engineering, you can produce a drug of a known molecule by imparting new property to it. Okay. So I myself was surprised that there are very many proteins which need such kind of upgradation. All these things, these molecules, they are in clinical use, approved, and if you can improve their properties, either half-life or stability or any other desirable property which is needed for particular pathological condition, then that can be a small little contribution. Coming to what is the mechanism by which proteins are cleared from the body once they are injected into the system. Renal clearance is a major contributor for removing the protein from the circulation, hepatic clearance, proteolysis, and endocytosis. So all these mechanisms contribute in removing the protein from the circulation. As a result, they have a low half-life. By knowing these mechanisms, one can tinker so that the engineered molecule can avoid one or many of such mechanisms and can circulate in the body for a relatively longer period of time. So this cartoon shows the glomerular filtration in kidney, which is a very normal process. Smaller the molecule, it can filter out. If the molecule is higher, relatively 60 kilodalton is a filtration limit, then it can patrol a body one more time till it gets degraded. So if we can increase the hydrodynamic radius of a protein which is getting filtered out by renal filtration, then we can increase its circulatory half-life and which will help in improving the pharmacokinetics properties. Okay. So variety of approaches are used to improve the pharmacokinetics of protein therapeutics. Glycoengineering, all these are proven, and many products are already available in the market. I will share with you. By engineering carbohydrate entities, conjugation with polymer, pegylation is there, and the chimerization is coming up lately, replacing the pegylation or issues associated with the pegylation. So glycoengineering of protein biopharmaceutical, one can take an example of daribapoetin, where it contains 2 additional N-glycosylation sites and exhibit 3-fold extended half-life as compared to its unmodified counterpart. It has to be administered less frequently as compared to unmodified counterpart. So these are known approaches one can increase the half-life of target therapeutic proteins. Alonva, it contains 28 amino acid long C-terminal peptide, which provide 4 glycosylation sites, and that helps in increasing its half-life. Second category is by conjugating with polymers. Pegylation has a long history of more than 20 years as a half-life extension module. Many pegylated proteins and other entities are already available in the market, and it is really considered to be safe and well tolerated. So many pegylated proteins are approved for the therapeutic use. These are some of the examples, and there are many, many more. We all are aware of contribution of this pegylation conjugation chemistry in bringing new drugs to the market. However, lately it is realized that pegylated proteins also have the problem, and they have certain limitations. Alteration in drug activity has been noted in many of the cases. Vacuolization in organs, different organs, continuous intake of pegylated drugs. In some patients has been reported that it accumulates in the organ, and in some cases immunogenicity has also been reported. Interestingly, 25% of us already have anti-PEG antibody because of use of PEG in very many other entities, what we eat or come across. So realization of this limitation advances this field of chimerization over pegylation. Here cost and time for conjugation chemistries are eliminated. Chimeric protein can be made in a single pot reaction. So this is a protein, engineered protein. Only a single round of purification is required rather than purification followed by conjugation and repurification of pegylated entities. And the half-life extension partner which are used in this, they are slowly degraded by natural process in the in vivo system. So these properties are encouraging, and then chimerization has emerged as a powerful approach to address the limitation of these therapeutic proteins. What is chimera? It's a mythological character. It's a fire-breathing creature and consists of 3 components. Lion, goat, and snake. Typical chimeric protein also consists of 3 components: your protein drug, your linker, and your half-life extension partner. So all these components and their orientations play a very important role in making one chimeric stable and functional chimeric protein. For the first component, you have more than 50 players available, which if you want, you can take it and try to develop a chimeric protein. With respect to linkers, linkers are very important entities. In our experience also, we found linkers play a very important role in structural and functional stability of protein, engineered protein. They are broadly classified into flexible one. If you want to keep the two domain apart, then you can have a flexible one. Rigid linker, which is a alpha helical signature, which Professor Park also talked about. He has used both flexible linker followed by two rigid linker in his engineered protein. If you want to keep the two domain separate, then you have to use the rigid linker, and then cleavable wrinkles are also exploited in certain cases. So you have a choice for deciding the linker. Third component is a half-life extension partner. A variety of them are available to design a chimeric protein. So you can take unstructured polypeptide, which can only increase the hydrodynamic radius and reduces glomerular filtration. You can take serum protein binding polypeptides to anchor your protein to the already circulating proteins of the body. Or you can take a domain of circulating protein itself, which not only increases hydrodynamic radius, but also facilitate FcRn mediated recycling. So all these are being used by a variety of peoples for developing their target molecule. Just to give you an idea, more than 13 chimeric proteins are approved by the authorities for variety of indication. Zyltrap, here a PEGF blocker for the treatment of cancer, developed by 2011 by Sanofi. FC region of immunoglobulin is attached to the VEGF binding portion from extracellular domain of this receptor 1 and 2. So this is a chimera they have developed, and it is working very nicely. Second example I would like to bring, dulaglutide and albiglutide, the GLP-1 agonist for treatment of type 2 diabetes. 2014 Trulicity developed by Eli Lilly, and 2014 Tenzinum by GSK. One point I would like to stress here, that the same GLP-1 analog is being chimerized with a two different half-life extension partner. In case of dulaglutide, it is FC region of human immunoglobulin, and in case of albiglutide, it is a human serum albumin. So just by changing the half-life extension partner, these two molecules are created and they are approved for clinical use. This is another example whereby chimeric proteins are used by using same entity, by using different half-life extension partner. So market is open if you want to use different linker, different half-life extension partner, then, and if the molecule is working, then you are in game. In this part, I will just share some of the research of my laboratory in a very quick session. Realization of that, we are developing 4 molecules for improving their properties. In first of the program, we are developing human arginase 1 as a broad spectrum anti-cancer agent. Arginine, we all know, is a versatile amino acid, play important role in metabolism. However, its metabolism is hampered in under certain pathological condition. If you deplete the circulating arginine by administering arginine hydrolyzing enzyme, then beneficial effect can be seen in those conditions. So arginine hydrolyzing enzymes have been shown to provide beneficial effect in plethora of conditions, not only in cancer, but arginase deficiency, immune system disorder, and other things. So they are the kind of hot molecules. Arginine hydrolyzing enzyme emerge as a strong candidate for treatment of variety of diseases. This is the mechanism for arginine oxotropic cancer particularly. These group of cancer, they cannot synthesize arginine and depend on body pool for their growth and maintenance. If we deprive the circulating arginine, then the cancer cell feel stress, and they make them more vulnerable. Either they die, or now they are prone to very many anti-cancer approaches like chemo or autophagy inhibitor or checkpoint inhibitor or apoptosis. So all these concepts are known, and people are developing particular formulation for particular cancer using arginine hydrolyzing enzyme. This is a normal cell. It need only, in this cartoon, 2 arginine. It can make 2 arginine. However, this is a cancer cell. They are voracious. They need lot of these materials. At the same time, if we deplete the extracellular arginine, then the cancer cell or tumor formation feels stress, and that is a point one can attack the cell with a variety of approaches. 2 enzymes, arginine deaminase and human arginase, they are undergoing clinical trials. Arginine deaminase is derived from mycoplasma, and ADI-PEG20 is the arginase, PEG arginase developed by Polaris group. They are studying under variety of cancer, as one can see. ADI-PEG20 received US orphan drug status also for treatment of hepatic cancer and invasive malignant melanoma. However, this enzyme we did not have at that time, so this enzyme was developed and taken further. There are limitations to this enzyme. The enzyme generates ammonia, results in hyperammonemia. Microbial origin also put the enzyme, make it highly immunogenic, and strong neutralizing response has been reported. Also, PEGylated version, they have the PEG toxicity and associated issues. Then comes the human arginase. It hydrolyzes arginine into ornithine and urea. It is a citric acid cycle enzyme present in our liver cell. Administration of recombinant arginase inhibit the growth and progression of many cancer. This has been proven. However, the enzyme exhibit low circulatory half-life. To address this, 3 versions are already in the clinical development. PEGylated recombinant human arginase variants are being developed. One of them is BCT100, which is developed by Biocancer Treatment International, Hong Kong, China. This molecule is bacterially produced and attached to the PEG 5000. This molecule received investigational new drug approval from FDA and is under clinical evaluation. Second player is pegxyl arginase, which is being developed by Aglia Biotherapeutics. This molecule is also bacterially produced, and they have cobalt instead of Mn, which is a natural cofactor, and is conjugated with a different chemistry. This is in phase 3 trial for arginase 1 deficiency and has been received rare pediatric disease and breakthrough therapy designation. So all these molecules are in advanced stages of development and approved for certain condition as well. Third is pectom arginase. This is also bacterially produced mutant enzyme using a different PEG moiety and PEGylation chemistry. This is reported very recently in the phase 1 trial. So this proves that engineered enzyme having an increased half-life can be really good. These molecules are being developed for variety of conditions because they show protective effect against a variety of cancers in preclinical and clinical studies. So recombinant human arginase is a broad spectrum anti-cancer agent that proves and can be developed for different cancer, those who respond to it. As I said earlier, all these competitors are PEGylated derivatives, and we claim that they have a PEG issue, the issues associated with the PEG entities, and we realize that there is a need to develop a safe arginase having improved circulatory half-life. So using protein chimerization approach, we are attempting to address this issue. This is a competitive landscape of our fusion arginase, and they have the advantage and disadvantage, and this is the current level. I will quickly just share some of the data, what we have done. We have designed, screen, identify lead variant. So designing a protein by de novo is a really a difficult job. You really do not know where to attach even one amino acid, or orientation or the combination can affect the structural and functional stability of protein. With the wisdom available at that time, we designed 30 variants using different half-life extension partner, different linkers, different orientations. 13 variants were cloned and expressed using E. coli expression system. And out of that, 4 variants were purified and compared in detail their enzymatic activity. From that, we got 2 lead variants reproducibly produced in our lab. We named them albuarginase and transarginase. Next step was to produce a lab scale production process for characterization. For that, we used a well-established PICIA expression system, which is used in the industry, and developed a protocol by which we can produce this enzyme in a pure and active form. After establishing that, we went ahead whether it is doing its desired job or not. We checked the pharmacokinetics activity, and we found that it is manifold higher than the unmodified arginase. So yes, our half-life extension partner attachment has increased its circulatory half-life. We are exploring the mechanism, whether it is FcRn or whether it is a renal clearance or both of them. We also checked its activity in vitro condition against cell lines, whatever we could get hold of, liver, prostate and lung cell line, and the purified recombinant enzyme is showing anti-cancer activity in cellular assay in a concentration dependent manner. We checked its activity in vivo cancer of SCC xenograph model, and this is the result. We found that combination of this enzyme along with the chemo, a known chemo, 5-fluorouracil, is more potent than either 5-fluorouracil or the enzyme alone. So we can, our idea here is to reduce the dose of chemo which is being used. If we can reduce the effective dose of chemo by half or 1/10 by using combination with this enzyme for particular cancer, then it will be great helpful. So this is the work done till now in this program. 2 leads identified. We got a clone, PICIA clone, although it is low yield clone, 2 to 4 milligram per liter. And we have a lab scale process which gives you purified protein and it exhibits the desirable activity. So that is what an academic lab at this moment can do. We are looking forward to take it further. Another molecule is in process of characterization. This is a prototype. This is a fish eye we always want to see. Develop our molecule, and the while we can send it to our collaborator so that they can check. We standardized its a buffer and condition whereby it can be stored for long duration of time and can be transported. Patent and papers we applied to protect the IP generated, and we are in process of that. This enzyme can not only be used for oxotropic cancer, but can also be used for variety of condition, as the competitor molecules are already being taken for these conditions. So once we can make this enzyme in large amount, we can check it in different condition as well. This is our dream. We are engineering variants of the enzyme. We will outsource this to produce it in the gram amount, and we want to see its effect and develop a beneficial effect in different disease model, and if that comes, we can take it formulation development for further studies. Our dream is to reach here. Let's hope how much we can do. Just couple of slides for other programs. In other program, we are developing peroxonase for nerve agent poisoning, where again using protein engineering approach, we have attached a non-PON protein to the peroxonase protein, and after screening many variants, we found the one variant. A lab scale process is developed, and this enzyme shows a pre-treatment of this enzyme shows prevention of OP poisoning symptoms and also mortality in animal studies. And the enzyme, engineered enzyme, also shows hydrolysis of G-type nerve agent. These are chemical warfare agent, and this is very high in demand, such kind of molecule for defense purpose. In last program, we are developing anti-TNF alpha antibodies for clinical use. We know anti-TNF market is a very, very huge market. There is a niche for everyone.